Hello everyone, I'm Dr. Deepthi Karya and today we'll discuss about the topic hemoglobin. Okay. Before starting my class, I would like to inform you all that you will get all my lecture notes in soft copy and hard copy. To get the soft copy, you have to download the application that is Vesalius and you have to get the course. And for hard copy, you can order my book that is Physiology Notes, which is available on Flipkart as well as Amazon. And link for the Flipkart as well as Amazon, they are given in the description box. Now, let us start with today's topic, that is hemoglobin. Okay, we all know that RBCs, they contain hemoglobin. So hemoglobin, that is oxygen binding protein which is present in our red blood cells. It is also known as coloring pigment and it is another name is chromoprotein. Chromo means coloring protein or metallochromoprotein. Why metallochromoprotein? Because here the metal is iron. Iron is the metal. Okay, and molecular weight of hemoglobin, adult hemoglobin, that is about 64,458. Now, synthesis of hemoglobin, erythrocyte precursors, they synthesize hemoglobin. You can see here, hemoglobin starts appearing in the intermediate normoblast stage. Okay, and mature erythrocytes, mature RBCs, they have hemoglobin, and but they cannot synthesize hemoglobin. Okay, hemoglobin synthesis starts in pro-normoblast stage. Okay, here, and it appears in intermediate normoblast stage. Okay. Now, what are the advantages of hemoglobin inside the RBC? Why hemoglobin is not outside the RBC? Because if hemoglobin is free, okay, outside the RBC, it increases viscosity of our blood. That is one disadvantage. Okay. Second disadvantage, because of increase in the hemoglobin in blood, it increases osmotic pressure of the plasma. And because increase in the osmotic pressure, fluid exchange between capillaries and tissue spaces, that is affected. Okay. Another disadvantage, that is if it is outside, this hemoglobin gets destroyed, rapid destruction by the reticuloendothelial system. Okay, and it is excreted by our kidney, that is known as hemoglobinuria, and that is found in pathological condition like in case of acute hemorrhagic or acute hemolytic disorder. Okay, when there is hemolysis, this hemoglobin appears in the plasma in free form and that leads to this hemoglobinuria. Okay, now you can see here, this is the hemoglobin, structure of hemoglobin. We will discuss in detail. Now, first of all, we will discuss normal hemoglobin concentration. Okay, according to age, normal hemoglobin concentration at birth, that is about 23 grams per deciliter or 23 gram per 100 ml or percentage. After 3 months, here this hemoglobin decreases and it reaches to 10 gram per deciliter. After 1 year, the concentration is about 12 gram percentage, and at puberty or after puberty, adult level is reached, that is 14 to 16 grams per deciliter. Okay, in adult males, in males always the hemoglobin concentration is more than females because RBC count is more in males. Okay, so this is 14 to 18 gram percentage or gram per deciliter, both are correct. And in females, that is 12 to 16 gram per deciliter or gram percentage. Now, how much oxygen is carried out by hemoglobin? 1 gram of hemoglobin can carry 1.34 ml of oxygen. Okay, so normal hemoglobin, if suppose we say that if it is 15 gram, so it will carry about 21 ml of oxygen per 100 ml of blood. Why per 100 ml? Because this 15 gram is present in 100 ml of blood. So you can say that oxygen carrying capacity is 21 ml, but because some amount of venous blood gets mixed, this becomes about 19.5 ml per 100 ml. Okay, and this is less in females because hemoglobin concentration is less in females. Okay, now structure of hemoglobin, as we have discussed, that is metallochromoprotein. This is conjugated protein. Metal, that is iron, iron containing pigment, and it consists of globular molecule, consists of globin chains. Okay, that combines with heme. What is heme? That is iron containing pigment. Okay. Iron is normally in ferrous state, Fe+2 state. Okay. And in abnormal condition, it becomes Fe+3 ferric state. Now, what is the structure of globin? First, we will discuss globin. We have four polypeptide, four globin chains. Okay. In adult hemoglobin is known as HbA. It has got two alpha chains. You can see here. These are two alpha chains and two beta chains. Okay. Alpha chains, it contains this one single alpha chain contains 141 amino acids, and beta chain contains 146 amino acid. Okay. And each globin chain is associated with one heme group. Okay. Now we'll discuss structure of heme. Heme is, as we have discussed, iron containing complex. Okay. So iron porphyrin complex. Okay, that is also known as iron and four pyrrole rings, protoporphyrin. You can see here, this is iron and four pyrrole rings are there. Okay, here. Now, this porphyrin nucleus, you can see porphyrin, it consists of, as I told you, four pyrrole rings. This is 1, 2, 3 and 4. Okay, four pyrrole rings are there. These pyrrole rings are connected with each other through this methane bridge. You can see CH, this one, methane bridge is here. Am I clear? Okay, then carbon atoms of the methane bridges, they are labeled as alpha, beta. You can see here, this is your alpha. I'll just show with other color so it becomes easy for you to understand. This is your alpha carbon, this one. Okay, beta, gamma and delta methane. Fine. Now, this 8 side chains are attached with the pyrrole rings. You can see here. This is 1, 2, 3, 4, 5, 6, 7 and 8. Okay. So here 4 methyl side chains. Which are the methyl at number 1 position? Okay, 1 position, 3 position, 5 and 8 position. We have methyl side chains. Okay. Then vinyl side chain. Vinyl, that is CH2 here. They are at number 2 and number 4 here. Okay. And propionic acid, that is CH2, COOH here. This one. CH2, COOH here. Okay. That is attached to 6 and 7 position. Okay. I repeat, methyl side chains, they are attached with 1, 3, 5, 8. Vinyl attached to the 2 and 4 and propionic acid to 6 and 7. Okay. Now iron. Iron, that is found in the ferrous form, as I told you, Fe+2 form. Okay. And that is attached to, you can see iron, that is attached to nitrogen terminal. Here you can see this one. Nitrogen terminal of each pyrrole ring. Here, nitrogen atom or terminal. Okay. On the Fe+2, a bond is available for loose union. Okay, here. And that is to attach or union with the oxygen. And when this binds with oxygen, this hemoglobin is known as oxyhemoglobin. And when it binds with the carbon dioxide, that is carboxyhemoglobin. Okay. Now, one molecule of hemoglobin contains 4 units of heme. Okay, and each heme molecule that is attached to one of the 4 polypeptide chains constituting globin. Okay, you can see here, one heme with one globin. This is heme, this is globin. Okay, so therefore 4 heme molecules and 4 globin chains are there. Okay, globin chains, and each heme molecule has one Fe+2 which binds with one oxygen. Okay, so you can say 4 molecules of oxygens, they are attached, or you can say 8 atoms of oxygen are attached to the hemoglobin. Okay, now we'll discuss functions of hemoglobin. First and foremost function, that is transport of oxygen from lungs to the tissue. Okay, in the lungs oxygen is attached with hemoglobin. This binding is known as oxygenation, not the oxidation. oxygenation. Why oxygenation? Because this binding is loose, rapid and reversible. Means loosely it binds rapidly and it easily releases when oxygen is required. And this is in the lungs. You can see Hb plus O2, HbO2. Okay. And at high altitude, another important thing, this binding is loose and rapid. One applied aspect here is when at high altitude, when 2,3-diphosphoglycerate increases, affinity of hemoglobin for oxygen decreases. Means oxygen is released easily from hemoglobin, and this is known as shift of oxygen hemoglobin dissociation curve to the right. I have explained this shift of right and left in my oxygen hemoglobin dissociation curve video. You can watch that one. Then carbon dioxide transport. Okay, carbon dioxide is transported from tissues to the lungs. Okay, this carbon dioxide from the tissue that is transported by combining with amino acid of the globin part. Globin is amino acid chain. Okay, so to this you have binding of the carbon dioxide. So CO2 plus HbNH2, this is the part of a globin chain and it forms HbNHCOOH, that is known as carbamino compound, carbaminohemoglobin, that is more readily transported than oxygenated hemoglobin. So this is why venous blood becomes more suitable for the transport of carbon dioxide than the arterial blood. Okay. Then third function of hemoglobin, that is it controls pH of the blood. Okay. Hemoglobin has 6 times buffering capacity than plasma. So hemoglobin binds with the H plus ion and it forms HHB. You can see here deoxygenated hemoglobin, which is not having oxygen. Okay, so this is when oxygen is been removed, this hemoglobin is Hb minus, so it binds with the H plus. So this is to regulate the pH. Another function is it has an additional nitric oxide binding site on the beta chain of the hemoglobin that is increased by oxygen. So hemoglobin binds with nitric oxide in the lungs, and releases it in the tissue. So this binding takes place in the lungs and this is released in the tissue. Where what is the role of this nitric oxide? It causes vasodilatation. Blood vessels are dilated, vasodilatation. Okay. Now we discuss about varieties of hemoglobin. Okay. There are two types of varieties. One is physiological and second is pathological or hemoglobinopathy. Physiological varieties are, one is adult hemoglobin or it is known as hemoglobin A. This one. This adult hemoglobin is again of two types, HbA and HbA2. HbA is the hemoglobin which is present in adult. Most of the hemoglobin is HbA and it has got 2 alpha chain and 2 beta chains. Okay, we have already discussed. And HbA2 that contains alpha chains and delta chains. No beta chains are here. Okay, this is minor component, only 2.5%. In we people also, we also have HbA2 but only 2.5% in normal adult. Okay, and this alpha and delta polypeptide chains are present. Delta polypeptide chain that is slightly different in amino acid composition than beta chain. Okay, here you can see how this differs. Only last 10 amino acids are different. As we have discussed, alpha chain has got 141 amino acid, beta chain has 146, and from this 146 only 10 amino acid sequence is different in the beta chain of, or you can see in the beta chain, and it forms delta chain, and that is HbA2. Third variety of hemoglobin, that is fetal hemoglobin, that is present in the intrauterine life, hemoglobin F. This is having alpha to gamma chains. Okay, and this is present in the fetal RBC, and gradually after birth of the baby, this fetal hemoglobin is replaced by adult hemoglobin. Okay, it has got alpha and gamma chains. Okay, fine. So easily you can remember HbA2, then delta, and fetal gamma. Try to remember this. Gamma means G and this is F. So you can easily remember alphabets G, A, B, C, D, E, F and G comes. So gamma chain, and here alpha and delta stands for D. So A and D. So this way you can make mnemonic and you can remember A2 has delta and fetal hemoglobin has gamma chains. Okay, here This gamma chain amino acid sequence is different from beta chain. About 37 amino acids are different. In delta chain only 10 amino acids are different. Here 37 amino acids are different. Now what are the special features of HbF? One very important feature, that is it has higher affinity for oxygen. So it will not allow the oxygen to release in the tissue. Okay, higher affinity means it binds with the oxygen and it will not allow it to release. That is one important feature. Second is, and this is beneficial for the fetus. So fetus has got more amount of oxygen consumption at low partial pressure of oxygen, because fetus is getting oxygen from maternal blood, not from the atmosphere. So it requires more oxygen. Then other characteristic feature is this fetal hemoglobin is resistant to the action of alkalis. It is not destroyed by alkali, and this is used in photoelectric calorimetric method, and we can estimate the HbF. Lifespan, disadvantage here is lifespan of fetal hemoglobin is only 1 to 2 weeks, whereas HbA lifespan, that is 120 days. Okay, so this is another difference between fetal and adult hemoglobin. This next is we will discuss hemoglobinopathies. These are the disorders of globin chain synthesis. Okay, here heme is normal, globin chain synthesis is affected. There are mainly two kinds of hemoglobinopathies. One is hemoglobin S and second is thalassemia. In hemoglobin S, we will discuss one by one, that is sickle cell anemia. Here what happens in sickle cell anemia, this is most important, most common hemoglobinopathy that is found in 10 to 20% of Negroes, and here sickle cell gene has been originated in the black population in Africa. And what is the pathology here? In the beta chain, 6th position of the beta chain, you can see here glutamic acid is replaced by valine. Okay, here you can see in the beta chain. So sickle cell RBCs are formed, and what happens here when sickle hemoglobin or HbS is formed, its solubility decreases at low oxygen tension. Okay, when oxygen tension decreases, this HbS is less soluble. So what happens? It gets precipitated. When it is not soluble, it is precipitated. It forms crystals, and these crystals you can see like this. They get elongated. Okay, and this crystals, they get elongated, producing change the shape of the cell, and the shape of the cell becomes sickle shape instead of normal biconcave RBC. Okay. Another thing is this HbS, that is less flexible as compared to HbA. Okay, and therefore it blocks the circulation. So sickle shaped cells increase. Another disadvantage is it also increases blood viscosity and decrease the blood flow. So sickle shaped cells, they are more fragile. Another disadvantage, and liable to undergo hemolysis, and therefore it produces anemia, sickle cell anemia, and this disorder is autosomal recessive disorder. Heterozygous individual with sickle cell trait, they have rarely severe symptoms, so that is recessive disorder. Okay, another important this hemoglobinopathy, that is thalassemia, which is also known as Mediterranean anemia, and that is due to decrease or absence of synthesis of polypeptide chain. Okay, these types, they are depending on whether it is decreased or absent, and which chain is decreased or absent. There are two types of thalassemia, alpha and beta thalassemia, and whether the chain is decreased or absent, there is thalassemia major and thalassemia minor. Okay, now so major and minor thalassemia, what are the differences between these two? Major thalassemia we will discuss. This is also known as Cooley's anemia or Mediterranean anemia. This is less common because most of the people having major thalassemia, they don't have long lifespan, so it is less common. And minor thalassemia, that is more common because the person is not having much more symptom rather than anemia. Okay, inheritance here, the individual is homozygous, means both the chromosomes having thalassemia gene, then the person is having major thalassemia. Heterozygous means only one chromosome has the thalassemia gene. Here the person is having severe to moderate anemia, where mild anemia is there in the minor thalassemia. Another, what is the pathology here in major thalassemia? Total absence of beta chain. Beta chain synthesis is absent, but here synthesis is reduced. Okay, and Because beta chains are absent, there is either synthesis of gamma or delta chain. And as I told you, if gamma chains are produced, then the hemoglobin is fetal hemoglobin. So fetal hemoglobin is increased. Here fetal hemoglobin will not be much more increased. Okay. Lifespan of major thalassemia child is less, may die by the age of 17 years. Here the person's lifespan is longer, and a person is having only symptom of anemia. Now, inheritance, you can see here, if the parents, they are, both the parents are thalassemia minor, okay, you can see here, chances of baby having thalassemia major are there. So always before marriage this thalassemia test must be performed to rule out that if one of the parent is minor, then and another parent is normal, then chances of baby having thalassemia are least. So this is, again you can see, this is the thalassemia gene from father and mother, and this is your autosomal recessive disorder. Now, derivatives of hemoglobin, that is reaction of hemoglobin. Derivatives means different derivatives from the hemoglobin are produced because of reaction of hemoglobin with oxygen, with carbon dioxide or hydrogen. So first is oxyhemoglobin, as we have discussed. When hemoglobin binds with oxygen, then it forms oxyhemoglobin, and this is, as I told you, rapid reversible binding. Okay, uh, whenever it is required, oxygen is immediately released. Then reduced hemoglobin, or it is also known as deoxygenated hemoglobin, when you can see oxygen is released, you can see here, when oxygen is released, then HbO2, you can see here, from which when oxygen is released, it is converted into Hb plus O2, and this Hb, that is reduced or deoxyhemoglobin. Okay. Third variety, that is carbaminohemoglobin, that is combination of carbon dioxide with the hemoglobin. You can see, as I told you, amino terminal of globin chain, it binds with the carbon dioxide and it forms carbaminohemoglobin. You can see here, this one. Fine. Then carboxyhemoglobin. Here, very important thing. This is carbamino hemoglobin plus carbon dioxide, and carboxy, that is hemoglobin plus carbon monoxide. Carbon monoxide, when it binds with the hemoglobin, that forms carboxyhemoglobin. Okay, COHB here. And the affinity of carbon monoxide is much more than affinity of oxygen. And because of this, oxygen from the hemoglobin reducing the oxygen carrying capacity of the blood. What happens when carbon monoxide gets bind with the hemoglobin? It releases oxygen, and therefore oxygen carrying capacity of hemoglobin is reduced. Another variety of this derivative of hemoglobin, that is math hemoglobin, that is HbOH. What is here? When reduced or oxygenated hemoglobin is treated with oxidizing agent like potassium ferricyanide, this ferrous form, Fe+2 form, that is converted into Fe+3, and this is your math hemoglobin. Okay, the sixth bond is attached with the OH, and that forms compound math hemoglobin. So math hemoglobin is written as HbOH here. Okay, so this is math hemoglobin. This Fe is converted into Fe+3. This is Fe+2. Okay, here. And disadvantage of math hemoglobin are, it cannot unite reversibly with oxygen. It cannot bind with oxygen. Okay, it is attached to the OH, that is not given off in a vacuum. So math hemoglobin, that is dark colored, and when it is present in a large quantity, that gives bluish discoloration, which is known as cyanosis. Okay, this also occurs due to deficiency of the enzyme NADH math hemoglobin reductase system. When there is deficiency of this system, because in we people also some amount of this Fe+3 is formed, but this system, you can see here, this is the system is NADH math hemoglobin reductase system. What does it do? It converts this Fe+3 into Fe+2. But when this is absent, this system, then this is not converted. Fe+3 is not converted into Fe+2. So we have increase in the level of Fe+3. So there is math hemoglobinemia. Math hemoglobin level increases. Okay. Another variety, that is glycosylated hemoglobin. What is this glycosylated hemoglobin? That is non-enzymatically binding of hemoglobin with the glucose. Okay, and this is also known as HbA1c. That is present in a healthy individual. It is present in very small amount, less than 5%. But when the person is diabetic and the sugar is not controlled, what happens? This sugar binds with the hemoglobin, and this remains in the circulation till RBCs are there in the circulation for 120 days. So if we found this value of glycosylated hemoglobin more than the normal value, you can say that this sugar control is not proper during last 120 days. So this is also one of the important diagnostic tests for the regulation or control of the sugar in the diabetics, and this gives us idea about the sugar control of last 120 days. This is another one. Then comes sulfhemoglobin. Here hemoglobin combines with the hydrogen sulfide. Okay, and this is, you can see here, when it binds with the hydrogen sulfide, you can see here this is SH group, and this is due to the drug like phenacetin and sulfonamide. When hemoglobin acted with this drug, and this sulfhemoglobin is formed, and when the sulfhemoglobin concentration increases more than 10 gram per deciliter, toxic effects they are present. Okay, so this is all about hemoglobin. Again, you can get all my lecture notes in soft copy and hard copy. To get the soft copy, you have to download the application Vesalius, and you have to get the subscription, get the course. And for hard copy, you can order my book, that is Physiology Notes, available on Flipkart and Amazon. Okay, thank you so much. If you like my video, you can like it, you can share with all your friends, and subscribe our channel.